Morning, everybody, and thank you for having us here today. My name is Esther Biswas-Fiss, and with me is my graduate student, Senem Cevik. And we're going to talk to you today about how we use the flow cytometry core facility through the voucher program to assess the presentation of normal and variant ABCA4 proteins on virus-like particles. Okay, so let's get started. What is ABCA4? I'm trying to advance the slides here. This new to blue genes. Okay, so we'll talk a little bit about what is ABCA4 protein to put this in a little bit of context, and that might help you because perhaps your protein is similar to ABCA4 and you can see some parallels in that. We'll talk about very briefly the genetics of ABCA4 linked inherited retinal degenerative diseases because that is what that gene is involved with. And we'll talk about how there are a variety of approaches that we can use to understand the pathogenicity of ABCA4 genetic variants and their impact on patients and predicting prognosis for patients. And then finally, the gist of this presentation today is how we've used flow cytometry to understand the membrane topology of this ABCA4 protein and its targeting to the membrane, which is really important in understanding certain types of ABCA4 genetic variants. And we'll just wrap it up with just some thoughts about the membrane imaging core vouchers and how you can use those to enhance your research. So ABCA4 is a gene that's involved in a wide range of inherited retinal degenerative diseases. On the left, we see the normal fundus photograph of the retina. And then as we go from left to right, we have increasing types of severity or different presentations, clinical presentations of the disease that is caused by genetic variations in ABCA4. Important for this particular group of investigators is Stargardt's disease, which is a early onset retinal degenerative disease that eventually leads to blindness, and usually that will take place, the blindness, in the late teens to early adulthood years. What is interesting about ABCA4 is that there is a broad range of spectrum of phenotypes and ages of onset. It is a progressive blinding disorder, and we really need to understand more about that because the ABCA4 gene has over 3,000 reported genetic variants. And some of these are frame shifts, some of them are missense, some of them are deletions or insertions, but that's a pretty large number. And we would really like to better understand the genotype-phenotype correlation to better direct patients to clinical trials, to better understand what's in store for patients who have not already been diagnosed with the disease, and to help genetic counselors tell their patients what might be in store for them. The genetic variants, this is a picture from Nomad browser, and it shows the ABCA4 gene, which is localized to chromosome 1. And you can see from this picture, and the little shapes indicate different types of variants, whether they're frame shift or missense or splice site variants, but they occur throughout the open reading frame. So it makes it very challenging to understand what's going on. There aren't any particular hot spots, for example, with this particular genetic variant. ABCA4 is a membrane protein, and I'll tell you in just a moment where it's localized, but it's localized to the retina, and it's in the rod outer segment discs. So you see up in the left-hand corner here that in nature, in the native state, ABCA4 spans the lipid bilayer. It has two extraluminal domains, and it has two nucleotide binding domains. ABCA4 is an ABC transporter, and like all ABC transporters, it utilizes the energy of ATP hydrolysis to transport substances across the membrane. When it has genetic variants, this can lead to loss of function of the transport, and therefore something will accumulate because it's not getting transported to where it's supposed to be. And again, this slide just emphasizes that there is a specific topology of ABCA4 in nature, and that the genetic variants are localized throughout the entire protein and to all its domains. So what does ABCA4 do in the retina? ABCA4 is involved in what's known as the visual transduction cycle. So remember, your mother told you to eat carrots, and the reason she did that is because beta carotene in the body is converted to 11-cis retinal, which acts as a chromophore for rhodopsin. And from your biology, you probably remember rhodopsin. And rhodopsin helps us convert the light signal to what we see as vision. And in so doing, 11-cis retinal is isomerized in the retina in these rod outer segment discs, which are found in the photoreceptor cells, the rods and the cones. It converts 11-cis retinal to all trans retinal. And therein lies the problem. So when the all trans retinal is produced, it needs to be then transported back out of the rod discs to be recycled subsequently in the visual cycle. And so here you see a lot of different proteins. It's a very busy slide, but essentially we need to convert the all trans retinal back to 11-cis retinal to act as a chromophore again for rhodopsin. So you need to have a restoration of the function, but equally important is that when this all trans retinal accumulates, it leads to the production of a cytotoxic compound called lipofuscin, and that leads to the death ultimately of the rod and cone photoreceptor cells. And so that's what we're trying to really understand is genetic variants. We know that they might cause a loss of function, and we also want to understand how aggressive that's going to be in terms of the toxic gain of function. And so what we really need to do then is to try to come up with a platform that we could functionally analyze these genetic variants because unfortunately not a lot is known. The challenge is that of those 3,000 variants that I talked about a few moments ago, about half of them are missense variants. And of those missense variants, over 60% are variants of unknown significance. So this is really troubling. We really need to get ahead of this so that we can, as I said, direct patients to ongoing clinical trials, come up with more precise therapies to relieve this problem. And so this is where our laboratory areas of focus comes in. We are trying to understand the clinical significance of these variants, determine which variants are pathogenic and likely pathogenic, according to the American College of Medical Genetics criteria, and which ones are benign or likely benign. So it's very challenging to work with the membrane protein. They're not as amenable to purification as non-membrane proteins are, but we really need to understand from a functional perspective what this means. We do use in silico tools as well, computational tools, but we have been working very diligently to come up with a in vitro platform. And so I'm now going to turn this over to my graduate student, and she'll talk about, we're going to get in now to what we've done to study ABCA4 and its genetic variants functionally, and how this relates to using the core facility vouchers for flow cytometry studies. Senem? We can't hear you, Senem.
Thank you very much, Dr. B, and thank you for reminding that I'm muted. Sorry, I'm also new to this platform. It looks more crowded. My name is Senem Cevik, and as we have a limited time, I will jump right into the topic. In our study, we utilize the virus-like particles, or VLPs, to express the full-length ABCA4 protein in the insect cell culture. And this system serves as a platform to produce the ABCA4 protein in a native light membrane environment, as Dr. B said, without the need for detergent solubilizations and reconstitutions of those conventional membrane protein purification methods. We used insect cells, but this system can be applied to any cell cultures like the mammalian cell lines. So VLPs are structures that closely resembles the viruses, but they lack the viral genome. They possess unique characteristics such as ability to separate the matrix proteins and bud off from the cell surface as host cell lipid bilayer. And this allows the membrane proteins on the cell surface to incorporate into the budded vesicles and be released into the culture media. One of our main interests was to investigate whether the ABCA4 protein, when expressed in this system, maintains its native-like topology on the membranes of the VLPs. Dr. B, can you proceed, please, with the next slide? Thank you. Now, I would like to quickly remind you the native topology of the ABCA4. ABCA4 has 4 functional domains. Excess cytoplasmic domain 1 and 2, ECD 1 and 2, and nucleotide binding domains 1 and 2. In the photoreceptor cells of the retina, the ECDs are inside the lumen, while the MBDs are on the cytoplasmic side, projecting outward into the disc structure. And we were curious if the same topological organization would be observed in our VLP system with MBDs outward facing on the VLP membrane structure. Since whatever on the cell surface goes onto the VLPs, the VLP maintains the same lipid bilayer composition of the host cell, as the VLPs bud off from the host cell plasma membrane as a one-way direction. So maybe we can show it. You can see it here. It's expected to be budded from the one-way direction. We would expect that the topological organization of those membrane proteins would reflect that of the cell surface. So to indirectly answer our question, we decided to examine the host cells, as it is easier to analyze the cells than the VLPs, and that's where we use the flow cytometry to determine the topological organization of the ABCA4 in the insect cell host cell plasma membrane. Can we move on, please? Thank you. So to determine this, we have again benefited from the flow cytometry, and very briefly, we used 2 domain-specific antibodies, anti-ECD1 for inside, which is normally an inside position in the native topology, and anti-MBD2, 2 specific domain-specific antibodies. And we also used a secondary antibody fluorescently labeled to stain our host cells that are expressing the ABCA4, and we did these 2 different stainings in different tubes. So we were hoping that if we could detect our MBD staining cells under the fluorescent channel, and we don't see any detection with the other antibody, anti-ECD1, that would just show us that the ABCA4 is a native and uniform topology in the cells, on the cell surface. We included various controls, such as isotype control, secondary antibody stainings, only empty VLP producing cells lacking the ABCA4, and then we used a lot of the watchers with multiple times of experiments. So if we can go to this other slide, which I want to show our results very quickly. Our results revealed that the only MBD2 stained cells were detected under the FITC channel, indicating the presence of ABCA4 in its native topology. If you see the graph showing the site scattered to fluorescence Alexa 488, which is the stained samples, you see there is no detection in the gated channel with the empty VLP or in the anti-ECD1, but we see that the cells are detected. They are Alexa 488 positive, therefore ABCA4 positive for those stained with the MBD2 antibody. We repeated this experiment multiple times, as I said, and consistently arrived at the same conclusion. In fact, if you see the bar graph here showing 3 different acquisitions normalized to the same number of gated cells, they clearly show that the MBD2 specific antibody staining showed the ABCA4 positive cells versus the ECD1 detection was statistically insignificant, indicating an unspecific binding. But this just tells us that the MBDs are outward facing on the cell surface membrane. So that was our conclusion. And I want to be so quick. So before I hand it over to Dr. B again to discuss more about our conclusions, I would like to take a moment to acknowledge Dr. Arit Ghosh from the Flow Cytometry Core Facility. And thank you very much for your time. That was it from me.
Great job, Senem. Great job.
Thank you.
Okay, so our conclusions, as she said, is that ABCA4 expressed in this heterologous cell culture system displays a uniform topology and that it's consistent with that of the native protein. And we found from this that flow cytometry can be a useful tool, and it can perhaps help us to understand other ABCA4 genetic variants that have difficulties with membrane targeting. Some of the variants of those 3,000 variants that you saw, their variant causes them not to target the plasma membrane or the rod outer segment disc membrane, and rather they remain misfolded and aggregate in the Golgi and the endoplasmic reticulum. And so it'd be useful to understand whether some of our variants are loss of function as a result of the failure to target the rod outer segment disc membrane. And in terms of the voucher, it was amazing. So this enabled us to get preliminary data that was used in an NIH application, which is under review currently. And it also was the cherry on the top of the sundae for a publication that's ready to go out shortly on this VLP technology. $4,000, which is the maximum amount of money that you can ask for, is actually a heck of a lot of time on the flow cytometer. We found that out. And so when you develop your application, you will work closely with Dr. Ghosh, because you need to come up with a budget and a research plan that he signs off on. So that's part of the application process. And even then, we sort of really had to work very diligently. I think Senem camped out in the core facility to get our work done in the time frame, because it is you should be mindful about the money you're asking for and about the time that it's going to take you to do these studies. And I thank you for your attention, and we would be happy to answer any questions. This is our lab group, some of our collaborators, and folks that are supporting our research. So thank you.